going to start the general session on global kidney voices. And we're going to talk about the role and some of the activities that are being done and looking at the role that patients are playing in leading the charge in driving global innovation and independence. We're going to have a series of speakers that will discuss different aspects of global kidney voices. And we're going to begin with Dr. Julianna Blum. She is a co-founder and executive vice president for corporate development for Humacyte, a good friend of AAKP, and we look forward to her presentation. Dr. Blum?
Hello, and thank you for the opportunity to speak here today. My name is Julianna Blum, and I am a co-founder and executive vice president at Humacyte Incorporated. I hope that you find the update I'm giving today invigorating and interesting, and one that aligns with the mission of AAKP. On my first slide, you'll see the disclosures and disclaimers for this presentation. All of the content is included in the text. At Humacyte, we are creating breakthrough innovations to improve patient lives and change the future of medical care. I'm excited to be here today to give you an update on our company, our technology, and the clinical progress we've made. Humacyte technology is being developed to support a platform of therapeutic opportunities across a broad range of clinical applications. Our first product opportunity, the human acellular vessel, has been evaluated for the past 10 years in opportunities like dialysis access for patients undergoing hemodialysis, for bypass for patients needing peripheral arterial disease repair, and for patients who have vascular trauma and need vascular replacement or reconstruction of their vasculature. As we move forward, we're continuing to explore the opportunities that the platform holds. We are now working on small diameter vessels for coronary bypass grafting and even pediatric applications like Blalock-Taussig shunting. We continue to develop our pipeline further and working to create complex tissues such as those that could be utilized as a biovascular pancreas, or even in the future, bioengineering organs. Humacyte has been developing and evaluating our bioengineered tissues for nearly 20 years now. We've spent a lot of time understanding the clinical need and creating products to support patient care. The organization was founded in 2004 by researchers out of Duke University. We are located in Durham, North Carolina. We moved our clinical opportunities, in particular clinical trials and dialysis access, in 2012, and have had the support of FDA through designations like Fast Track and the RMAT, Regenerative Medicine Advanced Therapy Award, since 2017. We've been able to create great relationships and partnerships with organizations like Fresenius Medical Care, the Department of Defense, and the American Association of Kidney Patients. More recently, we've had the opportunity to build out and fully validate a commercial scale production facility that can create our human engineered biotissues at commercial scale. Last month, Humacyte went public. We are now a company actively traded on the NASDAQ. The future looks bright, and I'm excited to continue the mission of our programs to bring our first product to market and develop our pipeline opportunities. Talking to you a bit about our human acellular vessel, the HAV, which is the same vessel we have been using in clinical trials since 2012. It's important to know we've spent many years developing our manufacturing system and creating an engineering system that can produce these HAVs at numbers that can support patients in need when the product is approved. Our bioengineering systems can grow batches of 200 HAVs at a time, and our current facility can make upwards of 8,000 units a year. When our facility is fully built out to capacity, it's possible to grow over 40,000 HAVs, or human acellular vessels, a year to support commercial launch and the opportunity for patients around the world to receive the product. These bioengineered HAVs are created using human cells to grow a new, robust vessel inside of a bioreactor bag. That bioreactor bag is inside the engineering system that creates 200 vessels a batch. These batches of vessels are created exactly the same way every time, resulting in a biological product that comes out of the box the same way every time a surgeon pulls it off the shelf. At the end of the growth process, we remove the donor cell identity from the cells used to grow these new tissues, package the product inside the sterile bioreactor bag, and ship those units directly to the hospitals. They're then available on the shelf at the time of surgery when the patient needs it. When the HAV is needed, the surgical team opens the box and removes the HAV. They're readily available on demand. I thought it would be interesting to show you a video of how simple and easy it is to remove the HAV from its packaging and have it available in the OR when the surgeon needs it and for the patients. The package is easily opened with the inside container being sterile. The solution inside the bioreactor bag is normal saline. The bag is opened, saline is drained, simple scissors can be used to open the thin plastic bioreactor bag. The HAV is then cut from the support system in the bioreactor bag and placed into a pan of saline. When the surgeon needs it, the vessel is ready to be implanted. Developing a bioengineered biological product allows us to address some of the shortcomings in currently available products. As you saw in the last video, the product is available off the shelf and immediately usable in the OR during the time of surgery. Over several years of clinical evaluation, we've seen that the HAV is also resistant to infection and has shown no evidence of immunogenicity when implanted into the patients on study. Probably the most remarkable attribute of the HAV is its ability to repopulate with patient's own cells, regenerating the HAV into the patient's living tissue. For end-stage renal disease patients, having an HAV available for access just 4 weeks after implantation is one of the benefits we provide patients undergoing dialysis access. In a little bit, I'll talk to you about long-term durability and the robustness of the product, as we've been able to capture some 5-year clinical data showing that the HAV not only continues to regenerate and support a living tissue, but also is durable and long-lasting. One of the key attributes and most important biological phenomenons of our technology is the ability of the HAV to repopulate with the patient's own cells. The HAV regenerates to become a living tissue. What if patients had the option to have something implanted that was as easy to put in as a synthetic graft, but had the biological components of a fistula? Once the HAV is implanted, the patient's cells begin to repopulate the structure. What goes into the body looking like a plain white tube becomes a living tissue. The image on the right is an HAV exposed during a routine procedure. The HAV has become a living tissue. You can see when the HAV is opened during the procedure that the wall of the vessel bleeds and is completely cellularized. The lumen is intact and shiny, demonstrating the ability of the HAV to repopulate and regenerate to become part of the patient's own living tissue. A regenerative therapy for vascular applications, such as dialysis access, has the potential to change the way we treat patients and think about their clinical care. Humacyte is developing this technology today. After many years of studying and evaluating our technology, we began our clinical trial programs in 2012. To date, we have 8 ongoing clinical studies across 4 different indications: vascular trauma, peripheral arterial disease, AV access for dialysis care, and a handful of patients who have been selected and approved by FDA for expanded access care. We have 460 patients on study in total across 60 sites and 6 countries. Throughout our experience, over 100 surgeons have taken and used the HAV clinically. With 5 clinical studies in vascular access for dialysis ongoing today, we have a good understanding of where the HAV would fit and benefit dialysis patients. As many of you know better than I, AV fistula is the preferred or gold standard of care for dialysis patients. And although this is best for most, it is not good for all. And there are many patients who are not suitable for a fistula or whose fistula never matures. For those patients, they have alternatives like synthetic grafts or catheters to use for dialysis. But those products, synthetic grafts or catheters, have shortcomings too and have complications associated with infection and durability. The HAV could provide a better access option for many dialysis patients, especially those who aren't suitable for a fistula or who are prone to recurrent infections. Our initial AV access studies were completed here in the US and Poland. There were 6 sites in total with 60 patients. Our first read on the study was at 12 months. Clinical data showed that the patency of the HAV compared favorably to what's been reported in the literature for PTFE and fistulas. We further evaluated different safety outcomes of the HAV as well, looking specifically at the biological attributes of the product. We saw no signs of aneurysmal degeneration. We saw no evidence of clinical rejection. And in fact, many of the patients on study went on to be able to have successful kidney transplants. This work was published in The Lancet in 2016. These studies provided us with clinical evidence that the regenerative properties of the HAV had a potential to improve patients undergoing dialysis care. One of the important impacts of the HAV being regenerative and becoming a living tissue within the patient is the ability of the HAV to heal after cannulation. For dialysis patients, cannulation is a way of life, and having cannulations after cannulations can be trying and challenging to an AV access conduit. For the HAV, we've seen clinical evidence of healing after cannulation. The 3 images in the slide show a new cannulation tract, show a cannulation tract that has healed to some extent, and at the far right, a cannulation tract that has fully healed. This is definitely an attribute of the HAV that is different than those synthetic or plastic types of accesses like PTFE. Looking at that same cohort of patients from the earlier slide, we have now followed them out 5 years. We have clinically demonstrated the long-term potential and functionality of the HAV. The image on the left shows a patient whose HAV has been utilized for dialysis for nearly 6 years. The HAV is durable and intact. Patency data from the cohort of patients still on study suggests that the HAV is favorable compared to fistula and PTFE values in the literature for patency over a long period of time. Following patients out on study gives us the indication that the HAV will be a valuable opportunity for patients for long-term dialysis care. Additional data that we've been able to analyze from clinical specimens shows that the HAV remodeling or regeneration is consistent across a wide range of patients, regardless of age, regardless of gender, or other attributes. We have seen histochemistry images that demonstrate that the HAV remodels to be a living tissue, regardless of the patient makeup. At Humacyte, we have spent a good number of years developing and evaluating our HAV product. In particular, the use of the HAV for dialysis access, PAD, and trauma. We continue to expand the engineering platform and developing new opportunities and initiatives to bring bioengineered tissues to a broader patient population. We're continuing to work on vascular opportunities, those in the small diameter space, taking an HAV and seeding it with islet cells so that perhaps one day it can be used as a biovascular pancreas to help treat patients with type 1 diabetes. And we've even begun unraveling the work around how to develop and create a bioengineered organ. Eliminating the wait list for organ transplantation in the future might actually be a reality. We hear the needs of the patients and are listening to the doctors and surgeons asking for better product options or therapies that can be used to fix unmet needs. The era of readily available tissues is here, and it's one that has an opportunity to change the way we think about medicine. Behind every innovation is a purpose. The road to success is not always easy or quick. It takes time and persistence to create and validate a new product for clinical use. Developing strong relationships is key, and it is definitely key to Humacyte's success. We value the partnerships we have with patient groups like AAKP, with industry and clinical partners, with payers and others that are helping to create the village that is bringing these readily available tissues to market. We are pushing ahead to finish our clinical trials and seek FDA approval. One day, our HAV may be the perfect opportunity for patients undergoing dialysis or other vascular needs. Our purpose is to provide innovative, engineered tissues for patients. We would like to extend an appreciative thank you to all of you who have supported Humacyte and our technology. In particular, we'd like to thank those who have participated in our clinical studies. Thank you again for the invitation to speak here today. It has been my pleasure to join you all.
Thank you, Dr. Blum. We appreciate your presentation and update about what's going on with Humacyte. And we look forward to continued collaborations with you and the organization.